Thank you, everyone. We're just going to wait 1 to 2 minutes before we start, just for anyone that's had a little bit of problems entering. So we're going to start. So first of all, I want to thank you all for joining in such great numbers. It really demonstrates how important management of Salmonella is. So today, crack the code and successful Salmonella control programs. Unfortunately, we've had a very last minute cancellation in one of our presenters. And so we have a switch out presenter today from the Philippines who has called in the last 24 hours to do an alternative presentation. Today's webinar duration will be 75 minutes. And the webinar is being recorded, and this recording will be available to everyone after the webinar. So it's really important today that we understand how important Salmonella is. We know that Salmonella is the leading cause of food poisoning globally, but it takes everyone to do active, proper surveillance and management programs for us to decrease this. It is an environmental contaminant in our production systems that causes extremely negative impacts on our health and also on our reputations. So today we're going to discuss the use of molecular typing for management of Salmonella. So the importance of being able to trace the strain of Salmonella in actively being able to determine the source and ongoing management decisions. We have 3 presenters today, and I would like to present our first presenter for today. With over 30 years experience in animal health and veterinary diagnostics, our presenter, Dione, has a master's in veterinary and human, animal and ecosystem health from the University of Pretoria, giving us experiences from South Africa. She has been involved in the Veterinary Laboratory Scientific Forum and specifically the AI or avian influenza task flocks, and more recently in setting up the guidelines for Salmonella management for that country. With all of this experience, she also has active experience in food safety along with animal health. And I believe that her input in today's presentation will give us some key indications of different steps we can take in active Salmonella management. She has been involved in ongoing lab management and ensuring that the conditions for ISO 17025 are met within the laboratories that she has been in. And in 2021, Dione joined On Point Diagnostics, where her technical expertise in Salmonella diagnostics has transitioned into assisting a range of laboratories in Africa in validating and implementing Salmonella testing and gaining their accreditation. Her experience and work follows the One Health outlook, where food safety laboratories and veterinary diagnostics she has consulted are dedicated to protecting the consumer health by monitoring active monitoring programs on the farm to fork approach to ensure that we can have sustainable, ongoing management for disease. So over to you, Dione.
Thank you, Alison. Today I will talk about how we've used Check and Trace Salmonella CTS technology in improving surveillance and decision making, primarily in the South African market. If we look at transmission of Salmonella, it is multifactorial. And one of the things we need to do is we need to prevent food from becoming contaminated. That is your end goal. And worldwide, chicken is your main source of Salmonella acquired infections, meaning that control programs within the poultry industry are essential. As Alison said, the Salmonella is one of the leading causes of food safety incidents worldwide. Unfortunately, in South Africa, we have very few actual government regulations, but most control programs are put in place by the producers themselves, as they are aware that they need to maintain a clean food chain. Next one. If we look at salmonella control in South Africa, historically, as I mentioned, there have been few government control programs. Food safety regulations require food to be free from Salmonella enteritidis and Salmonella typhimurium, while the Department of Animal Health requires that Salmonella enteritidis or Salmonella gallinarum in live birds is reported to them. The other place where there is a control is that imported meat must be free from SE, ST, SI, infantis, Salmonella Heidelberg, and Salmonella typhi. There is a draft program for import food that is exported to the UK, and of primary importance to our industry are Salmonella that cause disease in poultry, the gallinarum and the pyllorum. And bear in mind that less than 100 serotypes account for the most human infections. There are over 3,000 serovars, but less than 100 account for these human infections. And the most important ones that are transmitted from animals to humans are Salmonella enteritidis, Salmonella typhimurium, and Salmonella infantis. If we look at the common South African isolates, and you will find that around the world, different common isolates. Salmonella enteritidis obviously takes the top spot. We get a fair amount of Salmonella Heidelberg, Salmonella gallinarum, which is different to other parts of the world. We have a Salmonella gallinarum problem in our poultry flocks per se. It's not a human pathogen, but it is a problem for the birds. Salmonella hardar, Salmonella infantis, Swartengrind, Idican, and Seftenberg. If you look at the grouping, each one is a different group and the possible sources are very different. And this is where full serological identification assists the farmer and the veterinary team in finding the source of the Salmonella. If we look at salmonellosis in South Africa, in the 2023-24 year, which was recently published, we had 195 foodborne disease outbreaks. Of this, 50% of the pathogens identified in these outbreaks were non-typhoidal Salmonella. Many outbreaks were not investigated further due to insufficient epidemiological data and the absence of clinical, food, or environmental collection and testing. Unlike other parts of the world, as I mentioned before, Salmonella gallinarum is a problem in South Africa, and traditional identification of this isolate is very difficult. We look at our testing regulations. Our testing regulations are for the laboratories themselves. Food safety labs should be, and I say should, not must, should be ISO 17025 accredited. And the test method for isolation is not prescribed unless they are testing for imports and exports, and then they have to use ISO 6579. Veterinary labs must be Department of Animal Health approved and ISO 17025 accredited. And then the test method is prescribed. However, the labs are allowed to use a shortened ISO 6579, where the negatives are identified using Solus or Vitus, and then they continue on with the positives. However, serotyping, once again, is not a requirement for any of the labs. The consequences thereof is that the focus has been mostly on SE and ST, and many other serovars enter the food chain. With no clear guidelines for testing methods, this has led to often less sensitive methods being used. And informal slaughterhouses and backyard chickens are not effectively controlled. Very little, actually none, real-time testing occurs in outbreak situations. And this makes tracing back to source almost impossible. We look at traditional testing. If we follow the standard ISO 6579, back please, thank you. It traditionally takes 7 days. Methods like SOLAS, VIDAS, MSRV have shortened the timeline by 3 days, which has made a big impact on the laboratory's ability to report a positive result. And then Kaufman-White serotyping, which is your traditional standard serological identification of Salmonella, will take the laboratories a further 5 to 7 days. And if the laboratories do not have the serotyping capabilities, they have to subcontract, which then adds a further 2 to 3 days in getting the samples to a laboratory that does have the capabilities. From that, next slide. We look at CTS. Why would a lab want to then do Salmonella serotyping? If we look at the detection of Salmonella, just the species itself is insufficient to demonstrate a potential risk. And if we look at the latest standard that is not being signed off for our export of poultry products, it states that an entire farm is under quarantine and may not export if there's SE identified. But if you don't know what you have and you just have Salmonella species, you cannot then look at what your risk is and whether it needs to be quarantined or not. Different sources of Salmonella are often associated with different serovars, as I mentioned in the previous slides. And it's essential to know which serovar is involved to enable you to trace it back to the source to control the Salmonella. How do we use check and trace with that? A poultry company's Salmonella control programs protect both the consumer and the brand. And then quicker response times with CTS has allowed for birds to be treated before slaughter. And then sampling of hatcheries and feed mills to trace the source is now possible because there's time. And CTS serotyping identifies most of the isolated serovars. If you look at the 100 odd that are your primary cause of food poisoning, all of those are on the CTS list. And for labs that are in the veterinary field, Salmonella gallinarum identification is easy and quick, which makes life for your veterinary team a lot easier and quicker. Thank you. Next slide. So to say, if you look at this, when I said 5 to 10 days for your improved reporting times for your standard testing, if you use the CTS, it removes the Kaufman-White serotyping and you reduce your serotyping, your positive samples for day 4 to 5, depending on what method is being used, giving your farms and your producers time to trace back to source and to treat birds if they need to be treated before slaughter. Thank you, Leslie. In conclusion, and I have implemented the CTS in a few labs it's very easy to implement in laboratories with minimal training. The serotyping identification is objective and fast. And with many years of doing conventional serotyping within a lab, the plate agglutination testing is very subjective, and mistakes are made that much easier with CTS. The rapid result timeline of the CTS has assisted poultry producers in identifying serovars and controlling salmonella in their flocks. One of the major factors where this has been a bonus in South Africa is that imported products are kept in cold storage at the harbour, and for release, depending on what serovar or rework, and the time in cold storage, which is expensive for those importers, has been reduced. It has also managed the identification of contaminated products before entering the food chain. Thank you.
Thank you very much, Dione. I just want to remind everyone that we're leaving all questions to the end of the presentations. If you have any questions, please type them in the chat box and we will address them at the end of the presentation. And any questions that we are unable to answer due to time limitations, we will address in a follow-up to all registrants. Next, I would like to present our next presenter, very last minute. Abby Cordoa from the Philippines is presenting her presentation, Cracking the Code on Salmonella Serotyping in the Philippines using Check and Trace Salmonella. Abby is currently the Environmental Health and Safety Manager at Bioassets Corporation, and is also involved in the quality management in this service testing lab. Bioassets have been involved extensively in doing some serovar typing across the Philippines, and she will be sharing her experiences in doing this. So thank you very much, Abby.
Sorry, let me fix my audio and screen.
So,
hello, can everyone hear me? Is my audio clear?
Yes,
Hello?
yes, we can hear you.
Sorry for the internet connection. Can you hear me clearly?
yes, Abby.
Yes, okay. So thank you, Indical, for giving us this opportunity to present. And also, we'd like to share with you, as mentioned by Alison, our salmonella serotyping journey in the Philippines using the check and trace salmonella, where we like to crack the code. Uh, so to give you an overview of what will I discuss, I'll give you an overview of who bioassets is and salmonella as a livestock and public health concern, not just in Philippines but globally, and how our company use check and trace salmonella as part of our precision diagnostic initiative. So where we established two studies that I will be presenting today and another ongoing research study. So at Bioassets, we are a life science company situated in the heart of Batangas. So what we do and what we I hope to share with you today is how we support and foster partnerships and cooperation to address important problems in zoonotic diseases in our country. So we believe that uh, path to better diagnostic tools, uh, vaccines and policies and prevention starts with better information. So with that, as you can see in our pipeline, it is important that we have information wherein we discover, we detect, there's an evolution when it comes to different information at hand, and also the diversity. And from there, we will develop an application wherein we can use it in diagnostic, therapeutics, vaccine development, and warning systems. I heard from uh, the previous presentation that this tool can really help in safeguarding our response in future outbreaks. So, with that, we can have an outcome wherein we can protect, prevent, mitigate, and also support productivity. So, as you can see, we identify different gaps in our community. So, how can we respond to different outbreaks or zoonotic diseases at hand? How well do we know the enemy? And is there safe or effective vaccine on different zoonotic diseases in our country? So, with that, we gave us we happen to think of solutions like in responding to this diseases, we built a mobile biocontainment lab wherein we believe that there should be a point of need diagnostic responsible for faster diagnostics. And then we got the precision diagnostic and also the precision vaccinology. So the precision diagnostics here, as part of our precision diagnostics, we adopted the check and trace salmonella since we believe that truly identifying the enemy, not just the common serovar, but rather the serovar as a whole in determining and control, in gaining or making a good control program. So here is our research pipeline, uh, and today I will focus on salmonella. So as you can see, it's not just um, a problem in animal or livestock where we are into. We are a veterinary laboratory, by the way. Uh, it also impact humans as well. So again, salmonella is a livestock and public health concern, as we all know. And uh, with that, we can see that there are 80 million cases each year globally with these diseases. And there are increasing cases here in the Philippines, wherein 47 deaths were also noted. So this is a 2023 case. Uh, we are yet to find out that our 2025 data, 2024 and 2025 data. So increasingly, there are many cases here in the Philippines undetected. So what do we do? What did we do to help in this uh, salmonella problem? So first, we adopted the technology, validated it, and able to establish that it can replace the Kaufman White. So with that, we made a collaboration with the University of the Philippines, Los Baños undergraduate students on BS Biology major and microbiology program, wherein they they map 16 retail stores, egg uh, egg retail stores here in in Batong Malaki, Los Baños, Laguna. So in one locality. So from there, they got a sample of egg, and then uh, we employed microbiological and biochemical tests using these samples. As you can see, we uh, employed eggshell rinsing. After that, use a selective enrichment, specifically Rappaport Basiliadis broth, and then stick it on the loop full of it in XLD and HEA plates. So after that, those presumptive salmonella were characterized using biochemical test and then confirmed by PCR and then sent to our lab for serotyping. So 4 of their isolates, So they have many isolates, but then again, we just use four isolates to confirm or to identify its serovar. So as you can see, I have a control. I have a Salmonella typhimurion and Abetuba sample, and then this S5, S6, S8, and S9 were therefore isolates, and it was identified to be Salmonella antibed. So aside from this, we strengthen the Salmonella serotyping by employing a whole genome sequencing. So in our laboratory, we also do whole genome sequencing. So from there, the same samples that were used in the check and trace Salmonella were subjected to whole genome sequencing using ONT technology. So here's the process. We have the DNA extracted, purified, and then subjected to rapid sequencing DNA protocol, and then sequence it using fungal flow cell using fungal adapter. And from there, we identified that the salmonella that we have is really Salmonella enterica. So as you can see, the identified serial bar in the using CTS was supported by the whole genome sequencing that we have done. And with this, we also saw two plasmids in the whole genome, and it was further research on by some of our students. So next, aside from that, we expanded the use of the check and trace Salmonella technology by using it in different isolates from Salmonella enterica isolates from wet markets and abattoir in Metro Manila, Philippines. So we partnered again with UP Diliman, University of the Philippines Diliman, to better use the technology. We really want more and more people recognize that serotyping is indeed useful, can be a useful tool in um, disease and researches on salmonella. So as you can see, we revived 110 salmonella isolates from their 2018 to 2019 study from meat samples in abattoir and wet markets. And then we subculture it, stick it overnight. So we basically we revive it, suspended it in tea buffer for DNA extraction, and then those that are for CTS, they we revive it in a nutrient agar and submitted it to our laboratory for determination of the serovars. The other the DNA extraction was for PCR confirmation. The other path is for the multiplex PCR of the virulence genes and AMR genes. So basically, we just wanted to know if there is a correlation between, or there's a relationship between the Salmonella serovars and the virulence and resistance genes that we can we would know from BTEC and also the PCR. So from there, we will do data and statistical analysis. So this data is from our 2023 update. So we haven't updated yet uh, on the BTEC AST. But as you can see, the 110 has already um, have their virulence genes identified, their AMR genes identified. Now we're just waiting for the antibiotic sensitivity testing of this 110. But as you can see, uh, from this figure, this is the serovar distribution across the 110 isolates that we have revived so far. So as you can see from this pie chart, the noted Salmonella serovar from those samples is Salmonella recent, gaining 24% of the 110 samples, and then Salmonella annatum and Salmonella 145 12. I- So from this data, we will see if there is correlation between serovar and the area margin and virulence rates. So this is still ongoing research study. What I hope to emphasize this afternoon is that we really support uh, uh, salmonella serotyping in our country. Right now, I'd also like to share with you the current bioasset research collaboration using CTS in poultry industry. So we all know that uh, chicken and egg-derived products is the primary vehicle for salmonella infection. And as you can see in the country like Philippines, we really love eggs. Yeah. So we then built a collaboration with different research uh, and also research group and also one of the big farms here in the country to conduct a salmonella surveillance in the egg production chain. So as you can see here, we have UP Diliman Pathogen Host Environment Interaction Research Laboratory and the microbiology group of the UP University of the Philippines, Los Baños, and also our partner uh, farm, Batangas Egg Producers Cooperative. Uh, Here with from the team of bioassets, you have our president, Dr. Homer Pantua, a drug discovery scientist, and Miss Sinbay by um, our research director. So we built uh, this, just a disclaimer, bioassets. Department of Agriculture and Livestock had supported independently this study on surveillance. So we have different parts. This is a big study, and we supported one part of this study, which is on the serotyping. So this is still ongoing. The samples are currently still uh, processed, and we are yet to uh, find or find out the different serovars are yet to be identified. So as you can see, we wanted to really promote that salmonella surveillance in egg production chain is essential to determine where did this salmonella came from, or is there a crucial salmonella serovar that we want to control in the facility or in the farm. So we have different farms. Actually, even we have partner backyard farms in this study as well. So not just big or corporate farms, we also have backyard farms in the loop. So we wanted to have this prevention and control framework wherein we wanted to uh, identify or detect where the salmonella, and by doing so, we can establish yung prevalence rate, transmission route, and pathogenic potential of the identified serovars. So, yeah, we believe that it is important that we detect, we treat, we identify, track, eliminate, and continuously survey where the salmonella is. So, with that, thank you so much. Uh, we are now in sample collection, serovar identification, and genomic characterization. And we're heading to having my slides is automatically getting into the next one. I'm sorry. So we're heading on development of a control framework for our country as well. So thank you so much for this, for your time.
Thank you so much, Abby, for your presentation. And I think it really shows how important it is serotyping in the overall understanding what's happening at a country level from both South Africa and from the Philippines. Now we have had this. Our next presenter is Antonio, who is our head of business development for Check and Trace. But I want to remind everyone at the end of Antonio's presentation, there will be an opportunity for you to register to have one of our specialists contact you for a 15-minute discussion and discussing how the check and trace system may assist you in your country. So please hang in there. And also, any questions, please type them into the chat box. We will be addressing all questions at the end of Antonio's presentation. Every question is important, and we know that salmonella management is critical. So we're looking forward to your presentation, Antonio, on how this has been used within integrators for tracing infections within operations. Thank you.
Yes, thank you, Alison, and welcome everyone, and thank you for logging in today and taking the time to join us remotely. Yeah, my name is Antonio Sanz, and for the last 5 years, I've been working on the diagnostics of salmonella and the track and tracing of contamination. And today I will be talking about our tool, Check and Trace Salmonella, which able people or decision makers to support management decisions and to reduce the impact of salmonella. So let me start saying that traditional investigations of salmonella SPP often are not enough to on their own to effectively control its spread, and I'm talking about poultry companies, but also food processing companies. This means that interventions may come late or be targeted at the wrong points in the chain, leading to limited impact of the control actions. So to understand how salmonella moves from farm to fork, we need to know the type that is causing the contamination. So in this case, we found September and Infantis, and this is a real case on the slaughterhouse. Well, what you do is you try to trace it back to the source. In this case, we went through the broilers farms and we found still both salmonellas, September and Infantis. And we continue the tracing back to the hatchery and the breeder. And through the testing and sampling, we find out that we have two different salmonellas contaminating the production chain, which was Infantis and September. In this particular case, Infantis was coming from the feed, and September was coming from the breeders, vertically. So the decision making was to focus mostly on the feed to eradicate the salmonella, and of course go back to the breeders and make sure that all the breeder production was free of salmonella. Leslie, please. So a good typing method help us to understand the epidemiology of the salmonella, tracing the source of outbreaks and monitoring the effectiveness of these control measures. What I show you here is when you do serotyping in time, during a period of time, let's say 1 year, 2 years, eventually you find out that you will find the same type of salmonellas in the same farms and coming from the same sources. And for instance, also here is an example of a real poultry company which find out that, for instance, M. Vandaca and September in this case were always coming from the feed mill. So if a particular serotype is found in poultry feed, it may indicate that the problem in the feed supply it's the problem rather than at the processing facility. And so this information help you to pinpoint the source of contamination, and you can trace it from the beginning to the end of the productive chains. So that is a very important piece of why you need to serotype contaminations in general in all the production sites. Leslie? And this is another example, what I call yeah, fingerprinting. This is an example for one of our customers who was also using Check and Trace Salmonella to fingerprinting what was going on in their operations. It's very important to understand what is it that you have the weakest links to be able to take the correct actions, and not maybe cleaning or vaccinating all the facilities, but just focusing on the weakest links, which are usually always the same. Um, yeah, so the production chain we know is large, and each part could have a different impact in the carcass and in the final product. So knowing each serotype is present in each part of the production chain is essential. Why? Because different salmonella types have different levels of virulence. For instance, we all know that Salmonella infantis is much difficult to eradicate than others, so you will have to take different measures. We all know that Salmonella contamination can occur at multiple points in the production chain, or that different Salmonella serotypes may respond differently to sanitation methods. So that's why it's important to understand which serotype is contaminating your production chain. Leslie? So that serotyping information will lead to action plans. And that's the whole idea on understanding which Salmonella is contaminating your production chain. In this case, I'm showing a couple of actions that may be implemented after contamination of Salmonella in the production chain. So if you know that you have, for instance, M. Vandaca, Seftenberg, Orion, and Uacan coming from the feed, you just take measures there. If you understand that the hatchery may be the problem, then you have to take measures to avoid that contamination. Eventually, if the slaughterhouse receives salmonella-free birds, the slaughterhouse will be able to produce salmonella-free products. Leslie? Therefore, it's important to have a very good sampling program. And this has to be along the whole production chain, understanding what are the leakage links on the production, and eventually you have to understand that you have to sample to find salmonella. You cannot sample just to a couple of samples to just check if you have a feed product. You have to sample to find salmonella. Go on, Leslie. Yeah. So in this case, sampling helps to find and trace salmonella. Another click. Because salmonella contamination is mostly in a low number of samples and not evenly distributed, meaning when a flock is contaminated with salmonella, not all the aminas are contaminated, not all the overshoot samples are contaminated, not every ingredient of a sample is contaminated, just a part of there. Therefore, it's important to sample enough. And this, of course, this make sometimes our life uncomfortable, having to sample that much, but it's necessary when you, and then comes out to in the poison distribution table, meaning when you have, let's say, 10% of contamination in your production site, you have to sample less amount of samples to be confident that your salmonella, that your chicken production is free of salmonella. But when you have a low contamination rate, you understand that you need to sample far more to be able to be confident that you have no salmonella in your products. In this case, when you have 1%, you will have to sample far more along the whole production site to understand that you don't have salmonella. And this is something that we don't like to do, but it is necessary to be able to deliver a free product of salmonella and a fresh product. Go on, Leslie. So that's what I call intelligent monitoring. And for intelligent monitoring, you need to use all the advanced technologies and data analytics that we have in our hands today, including AI. When I talking to customers, they are saying that they are using Kaufman and White to type salmonella, or they just use it to, they do all grouping or just SA. I'm making understand that the Kaufman and White method is coming from the 50s. So we are now in 2025, and I believe we have better methods to be able to serotype salmonella. Another one, click on Leslie. Meaning, so, but you need to have that extensive sampling program, at least in the beginning when you are fingerprinting your production site. So you need, at least in the beginning, you need to have that extensive sampling program, understanding what is your production site about. So you need sometimes more samples than scheduled through the regulation, meaning you cannot only look for SA state. You need to understand if you have more salmonellas contaminating your production site. You also need, important again, sampling intended to find salmonella. And the typing is essential for tracing the source. Click, Leslie, go to the next one. Yes. So you may think that having an extensive sampling program costs you more. That may be in the beginning when you are starting to sample more, but not in the end. And what we see here is that in the end, that increasing of monitoring costs will leave you with benefits. Let's say that an average recall costs $10 million per recall, and I say in this for large-scale outbreaks or more. But if monitoring reduces that recall by 50%, let's say, it can save the company eventually millions. You can also improve sales because you get a little bit more confidence of the consumer. And if you boost your sales by 5%, that will mean a significant revenue growth. Or you are exporting to other countries, meaning that if you have a fresh product, free of salmonella, fresh product you can show, you can also sell for a premium price, which also improve your revenues. At some point, when you go from 15% salmonella positives to, let's say, 4.5 or less, that reduction of salmonella, that loss of value of the processed meat that you have to go through all different channels, then you can sell it as a premium product, giving you that benefit along the side when you are using that typing method for many years. That's what our customers usually never leave us. They are really loyal because they understand the benefits of using a Check and Trace Salmonella. Leslie? And this show, this is an example of one of our customers in Brazil. We started in 2015 with the typing of all presumptive positives in their operations. What you see is that the first year is not really showing a reduction of salmonella, and that's because you need some time for the actions to be effective. But after that, you see a clear reduction of salmonella in the whole production chain, their operations. It can never be going to zero. When someone say that I'm totally free of salmonella, it is pretty much impossible, because salmonella is always there, it's always present. But you at least can go a show here to 3 to 1%, what I'm doing in Europe, which is really a successful salmonella control program. And if you go to the next slide, Leslie, I will say that what is it that we are using. So Check Trace Salmonella, in this case, the 2.0 is a qualitative semi-automatic real-time PCR test, which is designed for the rapid confirmation and typing of presumptive Salmonella isolate. And it's developed to simplify the Salmonella identification process. And I'm saying this because we are able to type to give you a Salmonella name within 2 hours from an isolate. So you have like a hands-on time of 20 minutes and a run in the machine for another hour and a half. So in 2 hours, you will be able to identify the Salmonella, which is causing the contamination. And this has implications for mapping a manufacturing process and making faster to take the right decisions and focusing on pinpoint on the weakest links. Of course, we have accredited our platform. We have certified by Microval for the confirmation and typing of Salmonella, so that makes us equal and the only alternative for the ISO 5791 and say 5793 for Salmonella confirmation and typing. To do this with successfully validated according to the ISO 16140/6, meaning that if you want to implement at your lab, you will need to follow the 16140/3, which is really easy because we'll did all the work for you. You only have to run a couple of samples to be able to validate it at your own lab. The Microval certification, which is the ISO standard, is important, but we also require the AOAC certification, which is important for North America and South America. So we also have that. Go to the next slide. So we start, no, you go too far. Another back, one back. Yeah. Okay. So I missed one slide, but it doesn't matter. So we have 59 Salmonellas at the moment accredited, 59 Salmonellas, but it's an open system, meaning as we are speaking, we are adding another 47 serovars to the scope of the test, having in total a number of 107. And with those 107, we believe that we are able to detect at least 95% of all the Salmonellas that have been found in the last years in poultry production. It will be always Salmonella which are exotic. We have seen before, but with 107, we believe that we can cover 95% of what is being found. We are using 2 machines for that. One is already in the market, which is the BioRas EFS96 of the new Opus, and soon we have available a method working for the Quant Studio 5. Go on. Of course, there are challenges on implementing check and trace in any lab, and that can make the difference. What are the challenges that we have been encountering lately is that the control program based on serotyping may face challenges, including the need for specialized laboratory resources or the disruption of existing workflows. So you are used to the workflow, and the people in the lab is also not open to new technologies. That is important to understand, again, that we need the new technologies to be able to respond quickly to contaminations. Some labs lack the resources to scale testing when sample volume increases. That makes check and trace easy to implement, because when you have more volume, when you are testing more, you need to have also quicker outcome, and that help you our workflow, which is really easy. And there are also the issue of complexity, as some serovars may be more difficult to detect or more widespread, making control efforts than others. Go to the next slide, Leslie. Yes. What are the takeaways from the Salmonella control program based on Salmonella serotyping? Serotyping will allows you to identify the Salmonella and helping to trace the source of the contamination. That is the main reason why you need to serotype, because you can find two Salmonellas in the same facility, let's say one in the carcass and one in the bedding. And if you don't know you have two different Salmonellas, one in the carcass, one in the bedding, you may take actions in the bedding, not knowing that you have a different Salmonella in the carcass. So different serovars may have also different sources. Source tracking and traceability is very important. It's also a takeaway, because identifying which serovars are prevalent, and also if you have a house Salmonella, and a Salmonella which you can find now, when you find in 6 months, it means that you have a house Salmonella present in your production, meaning that you have to take different actions. It makes it difficult to understand why you have a house Salmonella. So tracking the contamination also is very important to pinpoint when it's occurring. At the end, the targeted control measures, control programs based on serotyping help to focus these interventions, these actions, where they are needed the most. Instead of cleaning all the farms, you may just clean the farm that is causing the contamination. So it is better to focus on the problem. And if a specific serovar is found to be a recurring problem in certain facilities or regions, then different prevention strategies can be implemented. And with this, I also come to an end to my presentation. I want again to thank you for your attention.
Thank you very much, Antonio. Right now on our screen, what you can see is we have a QR code which you can scan with your phone, and this will enable you to qualify for a free demonstration. So please book a 15-minute consultation with one of our experts, and we can discuss whether the check and trace technology will assist you in your control programs. In about a minute, we will start the questions. I can see a lot of questions have come through in the chat. Continue to add them. I'm not sure that we will get through the very large number of questions today, but I want to remind everyone that we will review all questions and respond accordingly. And that includes some of the questions that were sent in prior when you were registering for the webinar today. So we will get through what we can with our team of experts, and any other remaining questions will be addressed. We will put them into a document and we will email them out to everyone that has registered for today's webinar. So I really want to thank everyone for holding on, and we will start with some questions. And I will address them to our presenters today. So Antonio, because one of the last things that you actually presented is how common is it to find more than one serovar in birds at the same time, like a flock, a serovar, or multiple serovars?
Well, it's not that common, but it is possible. So we encounter several cases with our customers along the last 10 years, which shows that it's possible to find different Salmonellas in the same location. And it happens more when you have house Salmonella, meaning that you always will have that Salmonella. You're moving around the facility, but you encounter Salmonella coming from outside, let's say rats or people or different birds that may just contaminate your facility as well. So it's not that common, but it happens, and it usually happens more when you have let's say, this house salmonella standing in your facility.
Thank you very much for your answer. Just one quick next question, just for you, is that there's a question here around the turnaround time for the results being around 2 hours, but they've actually asked how long is the enrichment and what is the limit of detection for check and trace.
So we are testing from an already salmonella isolation. So the only thing that we do is just to pick up the colony from the agar. We boil it for 10 minutes to extract the DNA. We mix it with a master mix, and we pretty much put it in the machine. So it is really a short workflow. Depending on how many samples are you testing at the same time, and you can do up to 16 in one run. It will take you anything between 10 and 25 minutes. Again, it's just we are starting from an isolated colony, so it's just picking up the colony, boil it for 10 minutes, extract the DNA, and mix it with the master mix, and then you put it in the machine.
Perfect. Thank you so much for explaining that. Gioni, I think this question would be well answered by you. It has to do with how can the poultry industry balance the need for cost-effective salmonella control measures with the increasing pressure for stricter regulatory compliance and higher consumer safety expectations? I think in your current situation in South Africa, this is what you're aiming to address.
Thanks, Alison. Yes, the cost, I think when you're looking at how to balance the cost, it's a double-edged sword. The cost of your brand being affected by an outbreak and releasing salmonella into the food chain is way higher than actually doing the preventative testing of check and trace. What it does balance for you is the rapid response time, in that you have an answer where you can either stop your product going to shelf, leaving this, you can have a positive release system versus a recall system, which is more expensive as well. So it might, and cost-wise, comparing it to traditional salmonella serotyping, check and trace is actually slightly cheaper.
Thank you very much. Abby, I think you might be able to help some insight with this question. Would you be able to explain the advantages? Obviously, you're using next generation sequencing, but what are the advantages of the check and trace system over next generation sequencing in salmonella serotyping?
Hi, can you hear me?
Yes.
NGS is a powerful tool which give us plenty of information. But using CTS, it can deliver many serovar results in less than a day. So it basically, if we are into um, surveillance, which happens to have many samples, especially in poultry. If you have surveillance and you detect salmonella, it's very cost effective if we will use CPS rather than NGS, because of course there are expensive equipment, and also the turnaround time for results is also must be also considered in NGS. So with that, based on our experience, CPS is a practical choice when it comes to surveillance studies rather than NGS. Thank you.
Thank you, Abby. I can see that there's a lot of questions still coming in. I just want to remind people that actually in the chat box that we have actually also included the URL link if you want to book the 15-minute chat to talk about your detection. One of the questions.
Alison, I just wanted to say something because of the whole genomic sequencing. Of course, the complexity of the whole genomic sequence, which requires a specialized lab and stuff. Also, the time to filter all the information that system is giving you, all the data analysis makes check and trace more rapid in giving you a result and more easy and simple to implement. Also, the high cost of the sequencing, make the difference. Just wanted to say that. Sorry.
No, perfect. I think there's quite a few questions focused around what sort of sampling, what sample types, and when should I take them, is coming up around the general surveillance. So I don't know if Dione or Antonio, you have any insights into that part of the surveillance program prior to serotyping?
Yeah, maybe Dione can answer that question.
We're talking about laboratory testing here, to be clear.
Can I answer?
Yeah.
Sure.
Yeah.
Absolutely.
Yeah, on a surveillance study in poultry, specifically in layered farms, what we did was we have environmental samples. We considered environmental samples, egg samples, and also some. Actually, it will depend on your assessment initially. What samples do you need to? Based on basically what we did is we tour in the farm, get what can be the source of salmonella, and then from there we list all the possible samples where salmonella can be detected, so that you have a holistic approach in making the surveillance. So I hope that helps.
And also depends on the regions that you are in, depends of the type of production site that you have. And there are a lot of programs for sampling programs we can be used. Usually have to be led by a veterinary doctor that is focusing on your own farm and regions. But they are for sure sampling programs available that eventually could share. But again, this is different for every farm, for every operation.
And thank you, Antonio. I think that this is a critical part, that there is not a one size fits all. What we do know about Salmonella is that if we are actually able to detect the point of incursion, it allows us to implement changes that should reduce Salmonella moving forward. But there isn't a single answer to when I should sample. But what we do know is the more sampling and the more we are able to fingerprint our farms, it makes it easier for us to determine what actions we need to make in the future when we see similar strains. We have a lot of questions still coming in on that. I can see a couple of questions relating to the Quant Studio 5 version, Antonio. There's just two questions in there. One is, when is it going to be available? And number two is, will it be the same software for the Biorad system and for the Quant Studio 5?
So we are finishing the development right now. We are indeed now of the last stage of the development, meaning we are putting out the software learning, which we have to learn to understand and evaluate the curves that the qPCR is giving us, and translate that to a name based on the database. All developments are not always good with the deadlines, but we are hoping to have the system ready at the beginning of next year. So between December and next year, we hope to have something already working on on the QS5. The analysis platform in the cloud will be the same. The software will be different, but our customers won't see that. They will be using the same platform.
So thank you very much for your explanation and timelines. If people are interested in the CTS system for the Quant Studio 5, can they register their interest now? And we will let them know when it's available.
Sure, that makes all sense.
So I think we've also had quite a few questions in regards to vaccination. One of the things that's come up, and I think that this is a good question, is are you able to type vaccine strains currently?
I have that question many times, Alison, and let me see if I can give you the best answer possible. Yes, we are able to differentiate vaccinated strains from wild strains, but therefore we need the vaccinated of the vaccination strain sequencing. So we need information about the vaccinated strain to be able to differentiate it from others. If we don't have that information, that strain information, the vaccinated strain information, we are not able to differentiate. So if we were able to receive that information from the vaccine company, we will definitely be able to differentiate those strains. But first of all, we need that information. And to now, vaccine companies were not really open to share that information with us.
And so it's vaccine specific, right, Antonio? So one, it's vaccine specific, and for us to add it to the system, we need to have further information about the specific vaccine in question.
Yeah, correct.
So it is possible, but we need further information. So for that one, we need to look at a case by case. But I think Antonio's made it a bit clear that there may be some limitations on specific vaccines if not enough information is available. I think we have some more questions still coming in. I'm just trying to get through them, sorry. One of the questions is, does all Salmonella serotypes have protein signatures? So this relates back obviously to serotyping. So I'm not sure who would like to address that one. So serotyping is obviously based on cell surface markers. And so it is, but you may get some additional information when you're using a genetic method. And I think that's why we still detect many strains that aren't serotyped, but they are genotyped using the check and trace system. So Antonio, can you explain to me what happens when I end up testing for a serotype that is not currently in the CTS system? Will I be able to differentiate that serotype using this molecular method?
Yes, you will be able to differentiate the Salmonella. So we are targeting 27 genes of the Salmonella, and the presence or absence of those genes will give us a pattern, and that pattern will be translated by the software to a name based on the database. If we encounter Salmonella that we haven't seen before, or that it is a new Salmonella, or that it is not in the scope, then you will receive a Genova code. And that Genova code, which is based on letters and numbers, will be pretty much the same Genova code if you will test that sample many times. So if you have a recurrent Salmonella on the operations which shows the same Genova code, you will be able to translate it to a name using, in this case, whole genome sequencing of a reference lab in your country to be able to identify that Salmonella and that Genova code.
Yeah, absolutely. I have a pretty general question here. I think, what is the role of biosecurity in the control of salmonella control programs, and how does this relate to the use of serotyping?
Shall I take that, Alice?
Yes, please.
So the control of salmonella is multifactorial. Biosecurity is a huge part of controlling salmonella. If you don't have biosecurity, as Antonia mentioned before, you get outside contamination of your flock. If you don't know what you're placing, and what your feed looks like, what your breeder flocks look like, what their status is, you have a salmonella problem. So biosecurity goes hand in hand with finding out what salmonella you have. And although there was the question before about can you detect vaccine? Vaccination is an essential part of a salmonella control program. Whether you don't vaccinate or you do vaccinate, you just have to know what you're doing, whether you're doing it, and for what reason you're doing it. The serotyping comes in to, especially with check and trace, if you can rapidly determine which serovar you have. Certain serovars are linked to certain sources, not necessarily 100% definitive, but it gives you an idea of where to start looking for it. Some are more transmitted trans-ovary, and others come in as a feed source or an outside contamination source. I think that's about it.
Thank you so much for your answer. And I want to thank, firstly, everyone for participating, but especially our speakers today for their insights. I think, today probably raises a lot more questions, but one thing is clear. We need to do more in managing salmonella. We saw, how many infections there were in two, key countries, but this isn't limited to one country. This is a global problem, and the more action we take, the safer we will be. So I want to thank everyone. We will address the remaining questions, and there are quite a few, and we appreciate everyone's input, and we will share that with everyone. And if you need anything, please reach out to the Intercal team. We're here to help. And I hope that this was a valuable webinar for you around the management of salmonella. Thank you, everyone.
Yes, thank you, Alison.
Thank you.